Good day everyone! Welcome to another screencast here in Immunology and Serology. I am Dr. Sipache Basit, your lecturer for today's session. And before anything else, I'd like to greet everyone. Good afternoon, good morning, good evening, whatever the time that you are actually listening or watching to this particular screencast. Good day everyone! Okay, so for today's lesson, we shall be discussing the overview on immunity and immunization. What's the difference between innate versus that of the adaptive immunity? And then we will also be run through the different cells involved in both innate and adaptive immunity. Plus, of course, some of the most important organs that is vital to the functions of the immune system. Okay. So first, let us have some of this definition. Antigen. The word antigen is actually a short word for antibody generator. Meaning to say, whenever an antigen enters our body, so an antibody is actually produced as response to that particular antigenic stimulation. So let's take a look, for example, for coronavirus. SARS-CoV-2 is the name of the virus. So once the virus enters the immune system in response to that particular virus, which is considered to be as an antigen, so our immune system will be producing antibodies. Okay? So antigen, anything that is foreign, anything that is not recognized by our immune system is considered to be as an antigen. So which means that it is all around us in nature. Even the chalk dust, or if you have not been sweeping your floor, there's a chance that these particular dust particles may be considered to be as an antigen. Dust mites as well, these are considered to be as an antigen. So immunity is actually a term that refers to the condition of being resistant to infection. Okay? So if you are immune to a particular infection, which means that you are resistant, you will not get the infection. Okay. So the term attenuation refers to weakening, which means that it will make a pathogen less virulent. So there are several processes of attenuation, and some of them could actually take place by means of heating, aging, or even by chemical means. Such as, for example, formalin treatment, for example. Okay, so let's talk about innate immunity. Innate immunity refers to natural immunity. Therefore, this refers to the ability of our body to resist infection naturally. But when we say resist infection naturally, this would be by means of normally present body function. So, for example, the natural characteristic of our skin will allow us to naturally resist the infection. So innate immunity is considered to be as non-specific. So it means it is non-adaptive, which means when you say non-specific or non-adaptive, it means that the responses are the same for all pathogens or foreign substances to which one is exposed to. Okay, so that's what we meant by innate or natural immunity. So, adaptive immunity, therefore, on the other hand, is a type of resistance characterized by specificity for each individual pathogen or microbial agent and the ability to remember prior exposure. Okay, so in this particular bullet, let me emphasize two important terms. Okay, one is specificity. Okay, so when you say specific, a particular antibody is produced to a certain pathogen. Okay, specific to a certain pathogen, which means that if you have developed antibodies against the hepatitis B surface antigen, so the antibody is called anti-HBs, so that particular antibody would only work against hepatitis B, not with any other types of hepatitis. Okay, which means that if you have developed antibodies let's say against COVID-19. So the COVID-19 is the disease then the pathogen is the SARS-CoV-2, so the antibody would be specific to the etiologic agent of COVID-19, not with the previous SARS that happened way back in 2003. Okay, now, ability to remember prior exposure. This refers to an amnestic response. Okay, so when you say an amnestic response, okay, our immune system have the so-called memory cells. So memory cells have actually allowed us or allowed our immune system to remember prior exposure to the previous pathogen. And this explains the reason why once you have mumps, you develop a lifetime, or not really lifetime, but somehow a long-term immunity against mumps virus. So you might be wondering, I had several colds, episodes of colds. How come I am still having common colds virus every now and then, common colds viral infection every now and then? That's primarily because there are more than 100 types of cold virus out there. So that's why this particular cold virus will most likely infect you. Those virus that have infected you for you to develop common colds may be novel to your particular immune system. Okay, so let us discuss some of the cells of the immune system. Now, the main star, when we speak of the immune system, the main star here are the leukocytes or the WBCs. Of course, in hematology, you will learn that these leukocytes are already mature if they are found in the peripheral blood. So both these leukocytes would play both important role in our natural or innate, specific, or adaptive immunity. So this includes neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Okay, so this is an example of neutrophils. Now, in hematology, you will learn that neutrophils have a normal value between 51 to 67%, and in cases of bacterial infection, neutrophil increases. Eosinophil is between 1 to 4%, and if you see the orange-red granules, it is because it is made up of the major basic protein. We call it the MBP, which could be toxic for the helminths. And another thing that you have to remember is that eosinophil also increases in cases of allergic reaction and even parasitic infection. Okay, and then monocytes somehow can mature as macrophages, which is one of the examples of antigen-presenting cells. It is between 2 to 6%. And then basophil is the rarest type of WBC. If you notice, there are bluish-black granules that covers the entire cytoplasm. So basophil's normal range is between 0 to 1%. Okay, so as what I've told you a while ago, whenever monocytes mature or differentiated, so it may be differentiated into macrophages. So macrophages play a role in initiating and regulating innate and adaptive immune responses. So what are the innate immune functions? Believe it or not, but macrophages may be involved in microbial killing, anti-tumor activity, intracellular parasite eradication, phagocytosis, and secretion of cell mediators. Okay, so that is the function of the macrophages. Then mast cells, basophils will somehow mature into tissue basophil, so we call it mast cell. Okay, but they say that mast cells actually will actually just resemble basophil, but if we're going to check the lineage, it may not be the same as lineage as the basophil. So nevertheless, mast cells play an important role in allergic reaction, and they can also serve as the APCs. So when we say APCs, this refers to antigen presenting cells. So mast cells can enhance and suppress the adaptive immune response. Okay, dendritic cells are another examples of antigen presenting cell. So it is covered with long membranous extension that makes them resemble nerve cell dendrites. They are considered the most effective APC in the body and the most potent phagocytic cell. Okay, so we have been discussing APCs later on, and you will find out later what's the importance of these APCs. And then we also have the lymphocytes, between 20 to 40%. And guys, take note that there are two most important lymphocytes. These are the B cells and the T cells. Okay, so B cells are involved in humoral immunity. T cells are involved in cell-mediated immunity. Now, there is a particular population of lymphocytes that are not considered as B cells or T cells. We call them the natural killer cells. Okay, so the natural killer cells are also known as large granular lymphocytes. And what's the function of natural killer cells? Natural killer cells will actually lyse or have the ability to target cells. Okay, now remember, the target cells here in the context of immunology is different with that of the hematology, kasi the target cells in hematology refers to the Mexican hot cells or podocytes, but here in immunology, the target cells refers to cells that have been virally infected or cancer cells. Okay, so let's talk about the organs of the immune system. So first we have here the bone marrow, one of the largest tissues in the body, and the bone marrow fills the core of all long flat bones and the main source of hematopoietic stem cells, which develops into different blood cells of our body. This includes RBC, the granulocytes that we had previously discussed, monocytes, platelets, and lymphocytes. Okay, now remember, B lymphocytes have actually originated from the bone marrow, and the bone marrow is considered to be as the bursal equivalence. Okay, so remember, if you will be reading other texts, you will notice that there are two primary lymphoid organs. These are the bursa of Fabricius and the thymus. But since we are humans, we are not birds. Some may have birds. Okay, since we are humans and we are not birds, we do not have bursa of Fabricius. So what we do have, okay, we call them the bursal equivalents. Okay, now that we are adults, our bursal equivalent is bone marrow. But while we were still fetus, okay, the bursal equivalent was the fetal liver. Okay, so the bursal equivalent is the one that produces the B lymphocytes. Now, these B lymphocytes will, some of the lymphocyte population will mature and develop in the thymus. So they will migrate, mature, and develop in the thymus. So we call it the T cells. Okay, so T cells are small, flat, bilobe organ found somewhere here in our thorax, and it is the location for the maturation of T lymphocytes. Okay, B cells and T cells, both of them will eventually migrate to the secondary lymphoid organs. Okay, secondary lymphoid organs where the actions take place. Okay, because if you will be looking at the description here, it says that it functions as potential sites for contact with foreign antigens. So this is where the action is taking place, and increases the probability of immune response. Now take note, let's say for example, B cells and T cells that have not encountered any lymphocyte at all will be what? Will actually die by means of apoptosis. So they are called naive B cells or naive T cells. So what are examples of secondary lymphoid organs? This include your spleen, your lymph nodes, the MALT, which is also known as the mucosal associated lymphoid tissue, and the CALT, which is also known as the cutaneous associated lymphoid tissue. Okay, so spleen is considered to be as the size of the spleen is like the size of your fist, approximately like the size of your fist. The spleen, it is divided into two kinds of pulp. So we have the red pulp and the other one is the white pulp. Okay, So the red pulp, this is the splenic tissue that makes up more than 1/2 of the total volume. Okay, and it serves as the graveyard of the RBC. Once the senescent or old RBC reach 120 days, so this is where they are being destroyed. But don't be sad, because some of the components of the RBC, such as the hemoglobin, may be recycled. Globin in particular, and then heme will be degraded. So that's another lesson, okay, in hematology or even in clinical chemistry. Okay, and then the white pulp, okay, comprises about 20% of the total weight of the spleen. So if you will be looking at the structure here, okay, the white pulp contains lymphoid tissues arranged around the arterioles. Okay, so if you will be looking at the white pulp, so you will notice that it contains lymphoid tissue, okay, arranged around arterioles in, we call it the pulse. There you have it, okay, the pulse or the periarteriolar lymphoid sheath. Okay, so the germinal centers, where this is where the T cells literally will develop or germinate, for that matter. Okay, so this is the sites, the entire white pulp is the sites where you can find the T cells. Okay, so the spleen is also made up of primary follicles. Okay, so the primary follicles contain B cells that have not yet stimulated by an antigen. So we call it the naive B cells. Eventually, if they will not be able to encounter any antigen at all, means that they are useless and then they will die. We call that apoptosis. So nature dictates that if you're useless, you will die. Okay, lymph nodes. Let's talk about lymph nodes. Okay, so lymph node is the central collecting points for a white fluid that circulates within the lymphatic system. So we call it the lymph fluid or the lymphatic fluid. Okay, so the lymph nodes contain lymphocytes, and any other foreign substances enter nodes via the afferent lymphatic vessels. Though they will enter by afferent and exit through the efferent. Para hindi nyo malimutan, efferent exit, afferent enter. Wale yung joke na yon. Okay, so they enter by means of the afferent lymphatic vessels and exit through the efferent lymphatic vessels. Okay, now lymph nodes are, in Tagalog we call it kulani. So have you ever wondered, lalo na when you were still a kid, whenever you get infected, your kulani is actually swollen, or whenever the doctors are trying to physically check you, doctors will palpate something here, because the physician is actually trying to palpate your lymph nodes, trying to check if it's swollen, because most likely if your lymph nodes are swollen, then there's an infection. Okay, just like your spleen, lymph nodes may also have secondary follicles, and it consists of antigen stimulating proliferating B cells. It is location where transformation of B cells are actually taking place, and eventually B cells will mature as plasma cells and memory cells when they are exposed to an antigen. So they need to be exposed to antigen in order for the B cells to mature as either plasma cells or memory cells. So what's the difference? Plasma cells is like the antibody factory for body. So eventually, plasma cells will be secreting antibodies, and then memory cells are, as what I've told you in the introduction a while ago, memory cells would be important for what? For an amnestic response, to remember previous exposure to the antigen in the past. Mapagtanim pala ang memory cells kasi natatandaan niya kung ano ang nangyari nung nakaraan. Yun. Okay? So, we have also the MALT. MALT is the mucous. Okay, so I'm sorry for that interruption. the mucoid-associated lymphoid tissues, these are found on mucosal surfaces of the gastrointestinal tract, respiratory, and urogenital tract. So it is a main port of entry for foreign antigens, and it's numerous macrophage made up of numerous macrophages and lymphocyte present. If you're familiar with the oral polio vaccine, and even the vaccine against rotavirus, okay, so it has to be delivered orally. Why orally? Because those vaccines are made up of live attenuated antigen. So they will go through the gastrointestinal tract, and eventually the lymphocytes in the mouth will be able to encounter them. So we'll be able to encounter them, and that will stimulate mucosal immune response. And then the cutaneous associated lymphoid tissues are found in the T cells, monocytes, macrophages, and dendritic cells on our skin. Okay, so there you have it. The cells of the immune system. Okay, the introduction to immunity. So be sure to study very well for this particular lesson. Good day, everyone. Stay safe.